I'd like to welcome everybody to our Academy speaker series. This is, my name's Mitchell Lipp. You know me. I'm the chair of the Academy this year. And we're really, really delighted to have today's speaker because one thing that is increasingly becoming important in education is appreciating the connections between other fields, neurobiology and neuroscience, and cognitive psychology and education. Those silos, those barriers are breaking down rather quickly. In our own series, we've had in the past, we had James Zoll, who has been a pivotal figure in bringing these fields together and making neurobiology and neuroscience understandable to classroom teachers, and very useful thing. And ADEA, the American Dental Education Association, the theme of our next annual meeting is the science of teaching and learning, with a focus again on the contributions from neuroscience. So that brings me to introducing today's speaker, Wendy Suzuki, who is a professor of neuroscience and psychology at NYU. Her research focuses on two main questions: how our brains allow us to retain new long-term memories. And she's also interested in the effects of aerobic exercise on our brain's ability for learning and cognitive abilities. I think we have a lot to learn, and I'm really looking forward to today's presentation. So
Great.
Thank you for joining us.
Thank you. Well, thank you for that kind introduction. It's a real pleasure to be here. And you guys brought a change of clothes, right? Because we're going to do a little, no? Okay, well, that's okay. So I'm going to tell you about three things today. First, I'm going to give you a little overview about how memory works. What do we know about the basic brain areas important for memory and what happens as we form new memories? Second, I'm going to tell you about some of the recent research from my lab that uses single unit physiology. We're looking at the patterns of electrical activity in the brain as animals are learning new pieces of information. And we have insight, new insight into how new memories are actually being formed. And finally, the key question: how do I improve my memory? And I'm going to be telling you about some recent findings from studies that I've looked at using exercise, aerobic exercise, and asking how aerobic exercise can improve our learning, memory, and cognitive abilities. So hopefully you'll leave here with a theoretical understanding of neuroscience, but also some practical tips as well. Okay, so how memory works. The field of memory is quite unique because you can point to a single paper, one paper, published in 1957, and you can say this is the paper that started the modern understanding of how memory works in the brain. It was a paper authored by William Scoville, who was a neurosurgeon, and Brenda Milner, who's still alive and kicking and doing very well today at McGill University in Montreal, who was a neuropsychologist who examined and characterized memory deficits in a group of patients, and in particular one patient who's very, very famous. And the initials of that patient are HM. So this paper focuses on a structure well, this paper brought to light the importance of one particular brain structure in memory, and that is the hippocampus. Here I'm just showing you where in the brain the hippocampus is. You see the eyes here. This is the temporal lobe outlined in red. You have the hippocampus here, important for long-term memories, as I'll tell you, and I'll tell you why we know that. And then right in front of it is another structure that I'm not going to talk about today, but might be particularly relevant for dental kinds of things to kind of suppress. These are fear memories that can be associated with visits to the dentist, and they're actually quite important perhaps for you to understand because they are very pervasive. They're very, very strong kinds of memories. As you all know, memory for fearful kinds of things are sometimes hard to forget. And in fact, the kind of aberration of these kind of amygdala-based fear memories are what causes PTSD. We're not going to talk about that kind of memory. We're going to talk about the kind of memory that is dependent on the hippocampus. So I just want to show you that. Oh, I should also tell you that the word hippocampus means seahorse. And this is actually a dissected human hippocampus and shown right next to a seahorse. So you can see that the anatomist that did this wasn't drunk or anything. He actually had a good reason, he or she, actually it was probably a he, had a very good reason to call the hippocampus a seahorse. So it is this curly Q kind of structure that does look like a seahorse. Okay, so what happened in 1957 that caused Scoville and Milner to write this paper? What they did was, Scoville was what's called a psychosurgeon at that time. He was doing explorations, in fact, basically human experimentation to ask what kinds of brain lesions or brain manipulations could help resolve issues like schizophrenia, bipolar disorder, as well as epilepsy. Now, we knew from even before 1957, in the 1940s, we knew that if you removed the hippocampus, they knew where the hippocampus was, on one side in cases of severe epilepsy, particularly if you found the side where the epileptic focus was, you can relieve epilepsy. Now, so they might have done that. But this patient, HM, whose initials are HM, came in with such a severe epilepsy. HM sustained head injury at the age of 7, experienced his first major seizure at the age of 10, first generalized convulsion at the age of 16, and between the age of 16 and 27, these epileptic seizures got more and more severe. He was not able to hold a job. It was so severe he could not lead a normal life. This epilepsy was not alleviated by any of the drug treatments that they saw. It was a very, very severe form of epilepsy. And because of that, they decided on an experimental approach. They wanted to ask the question, well, if removal of one side of the hippocampus is good, because this case is so severe, why don't we try double removal? This is a serious operation. Let's go in and get both of them and make sure all of the epilepsy can be resolved so this patient can go back and live his normal life. You can see that this is the tip of the temporal lobe, and the lesion, they wanted it to go back about 8 centimeters from the tip of the temporal lobe bilaterally here. And this is a rendition of what it would look like. Here's a coronal section, a section like this, through more posterior, and you can see both sides are removed. And this is just what the normal brain would look like. Here's the hippocampus here and there. So that's exactly what they did. And the good news was that the epilepsy was severely, significantly reduced. There's a few chairs up here. If you want to come and sit up here, there's 1, 2, 3, 4, 5, like 6 or 7 chairs up here. So this epilepsy was significantly reduced. But the bad news was that immediately when patient HM awoke from his surgery, it became clear that his memory was severely and devastatingly impaired. Why do I say that? Nurses could come in the room and have a conversation with him and leave the room for literally 30 seconds or a minute, and he would not remember ever seeing that nurse. Now, could he remember other things? Could he remember language? Yes, his language was fine. He remembered everything that he learned in school. His general knowledge was just fine. He knew who his mother was. He knew who his father was. He remembered Christmases and growing up and his school friends. That meant that this removal does not affect your well-established long-term memories. It means that this area is not where long-term memories are stored. It means that this area was critical for your ability to put new information in long-term memory. Now, not HM, but since that time, unfortunately, there have been other patients that, by other circumstances, have come to have lesions in these areas. In particular, there was a case in San Diego, patient EP, that had encephalitis, and his encephalitis basically caused a lesion that looked very much like HM. And what these patients a tragedy of this situation is they know they have a terrible memory impairment. And they're in fact aware that it steals your life. I mean, what are you without the memories that you accrue every day? If you can't remember anything that happened over the last 5 years, are you the same person or a different person? So from the age of 27, HM could not form any new long-term memories. He could do things like do crossword puzzles. So it was very famous. He loved doing crossword puzzles every day. So he could learn and retain language, or he learned language and he could retain it. But he couldn't remember. In fact, Brenda Milner was a fixture in his life for many, many years, meeting him when he was 26 and studying him for many, many years. Did he recognize her? Well, actually, at the end, if pressed, he would say, yes, I do know you, but you must be somebody that I went to school with. So he had a small ability to remember and form new memories, but he could not remember any of the details. So what do we think, and what did we learn? And the reason why Brenda Milner is such a giant in this field and really kind of created this field of understanding memory is because she had such beautiful and elegant insights into this. So what she was able to understand from HM is that the memory impairment that was so pervasive in HM was not a global memory impairment. It was very severe, but she was actually able to show that there are certain things that HM was able to learn completely normally. What could he learn normally? He can learn fine motor skills. So the famous task that she discovered that he could learn something was a mirror reading task. So you'd have to read a whole passage on a mirror, from mirror, so it's mirror reversed. And so he had language abilities. And at first it was very slow. He had to work out the words just like you and I would. But with practice over days, he got faster and faster and faster at doing it. And you could measure his rate of learning, his speed of mirror reading learning, and it was completely identical to normal people. So that was amazing. He did have normal ability to do that. But the difference was, you ask him, why are you so good at mirror reverse reading? You're really good. And he would say, I just must be good at that. This is a talent of mine. And you would ask him, do you remember the episodes that you practiced? You practiced this for 3 hours yesterday and the day before. Do you remember that? He would say no. So what HM lacked was what we call episodic memory. Memory for remembering the episodes of our life. That's why it's so devastating when you learn it. You cannot remember the episodes. That's what makes up our own personal histories. But motor skill learning was completely intact in patient HM. So let me go forward. So several of the other key conclusions that Brenda Milner came up with from this classic case was memory, and this, what she means by this is episodic memory, memory for events in our lives, could be impaired selectively without the loss of other cognitive functions. He could pay attention to things just fine. He can learn other kinds of tasks. So many other cognitive functions were completely intact, even though his episodic memory was impaired. Second, this showed for the first time that memory was localized to a particular region of the brain. I emphasize that because you can ask, well, what did people think of before they knew about HM? Before they knew about HM, the field was dominated by a theory from a famous psychologist who worked at Harvard named Carl Lashley. Carl Lashley did other experiments in rodents and concluded that memory was distributed throughout the cortex. He did experiments where he showed that it didn't matter where in the cortex you damaged, but what mattered was how much cortex you damaged. So a lot of cortex, no matter where it was, would cause a memory impairment, but a little damage, no matter where it was, would never cause a memory impairment. We now know that those experiments were wrong because he was looking in the wrong place. The hippocampus is not in the cortex. It's deep to the cortex. So the prevailing view before this was you can't study memory by studying a particular brain area because it's very distributed. And people believe that because of the findings, the interpretations, and because we know memory is quite complicated. So the idea that you needed lots of interactions, that kind of made sense. So that's another reason why this 1957 paper was so stunning to the field. You mean Lashley was wrong? Yes, he was wrong. And the answer is memory is localized to the structures of the medial temporal lobe. They concluded that the hippocampus is a key structure. But the impairment, but there was a caveat, because in fact, HM, the removal included this part of the temporal lobe as well, which I just showed you included the amygdala. So they weren't sure whether this lesion was due to bilateral damage to the amygdala, to the hippocampus, or combined damage to the amygdala and the hippocampus, or bilateral damage to the amygdala alone. They actually didn't think it was bilateral damage to the amygdala alone. They had another case that had exactly that kind of lesion, had no memory impairment. But they had other cases showing that the more you went back, the more the lesion included the hippocampus, the more severe the memory deficit was. Okay. And I just put this in, initially they described this memory deficit as global, but later studies by Brenda Milner showed that it wasn't global, that it was specific for this kind of episodic memory that I was talking about. And as I said, HM could learn other forms of memory. So that is kind of how we got an introduction to memory. And now I want to jump into, well, what do we know further about memory, kind of the modern study of memory. And we were left with this question, is it the hippocampus alone? Is it the hippocampus plus the amygdala? And here, of course, you can't go and experiment on humans. One of the main messages of that 1957 paper is, look what you found, neurosurgeons, you cannot ever do this again. And so, of course, this wasn't done, and including Scoville, who was doing these experiments, including medial temporal lobe resections, to look at ways to alleviate, as I mentioned, schizophrenia and bipolar disease. And so that was not done anymore. And so what we moved to was experimental animal model systems. Rats have hippocampi and amygdala. Monkeys have hippocampal and hippocampi and amygdala. And so we can look at the effects of these specific lesions in animal model systems. And that's exactly what we did. And I'm going to jump and summarize all of these studies to tell you that the key structures that we now know are important for memory include not just the hippocampus, but the surrounding cortices that are called entorhinal, perirhinal, and parahippocampal. Where are these areas? They surround the hippocampus. The hippocampus is deep to this cortical area right here. Tube-shaped structure. This is the ventral view of a monkey brain. And these cortical areas, we now know, are also contributing to memory function. And in fact, we now appreciate that many of these structures, particularly the entorhinal and perirhinal cortex, were severely damaged in HM. While they focus on the hippocampus, they got damage to these cortical areas. And that was verified not just in an MRI scan that they did in HM later in his life. I should just say that I don't know if any of you remember on December 4th, 2008, on the front page of the New York Times was a picture of patient HM and his name revealed for the first time. So the general public appreciates the importance of this patient. And I remember, as a student for 20 years, studying all that HM kind of gave us in terms of understanding of memory. It was a very memorable day for me. I remember I was teaching undergraduates and I brought the New York Times in, but it was also very strange because it was the very first time that I had learned his real name. I had referred to him in my mind as patient HM. Of course he has a name. And more of his details. I also knew quite a bit about his background, but I never knew his name. He was a very, very generous person. He was never able to live on his own since the age of 27. He lived with his parents until his parents passed away, and then he was taken care of in a old age home. But continually throughout his life, from the time he was 27 into his 80s, where he started experiencing age-related cognitive decline, he was an enthusiastic participant in all of these memory studies, and had the realization, they've asked him, what do you feel about, how do you think about, participating in all these memory studies? It must be tiring. And he says, I hope that they can learn something from me. He just was very grateful or happy to be able to contribute, which I thought was very, very touching. So he has passed away, and you might be familiar with the fact that his brain has been donated to science, and they streamed online the sectioning of his brain, which is probably the most famous brain in all of neuroscience. By that time, I have to say that his brain had degenerated based on aging, but still a historic kind of thing. It's still, I'm not sure whether they're finished yet, or they're certainly not finished with all the staining, but his entire brain has been sectioned, and so we're going to be able to study the detailed anatomy of exactly what was going on. But now I want to jump to modern studies where we know exactly which one of these structures are important based on lesion studies. We can lesion these areas and get, both in monkeys and in rats, and get severe memory deficits that are selective to episodic-like kinds of memory tasks and not impairing other memory kinds of tasks. So it's a very good animal model. We know the anatomy. So this complex diagram is some of the anatomical connections that I worked out when I was a graduate student, showing not only the interconnectivity of these brain areas, which are the key to memory, but also presenting to you why these areas are at this level that they can contribute to memory. Memory involves all sensory modalities. It involves processing lots of different kinds of information. And look at where these areas sit. They're receiving convergent, strong convergent input from all of these other brain areas in the frontal lobe, other areas within the temporal lobe, insular cortex, important for somatosensory, cingulate gyrus, important for decision-making and other motor functions, and parietal lobe, spatial functions. So all of this information is projecting onto these cortical areas that then subsequently project onto the rest of the medial temporal lobe, and all of these areas are contributing to memory. But the question that I wanted to address in my studies was not just the effects of damage to these areas, where you have to interpret. So animals are impaired at something. What exactly was the area that you lesion doing in normal life? And so the approach that I chose was looking at the electrical activity in normal animals performing a task. This is called behavioral neurophysiology. So I can introduce very thin microelectrodes into the hippocampus or entorhinal or perirhinal or perihippocampal cortex in awake behaving monkeys and basically have them playing computer games. So in my lab, we have a lot of monkey-based computer games, memory games, set up in whole bunches of different rooms. Monkeys come up from their housing situation every day, and they get ready to play their computer game. And for reward, they don't get money. They get juice. They're thirsty. That's the only time that they get liquid reward. And so we also spend a lot of time optimizing that juice. So we're actually making fresh fruit smoothies for them, and choose, some of them like veggie smoothies, some of them like fruit smoothies. You'd be shocked at the range of taste preferences in monkeys. But one of the goals of all the students in my lab is to find the superfood for their monkey, what their monkey loves, and reserve that for when the task is hard hard or you really need to motivate the animal. For some animals, it's tomatoes. They would die for a tomato. Other animals hate tomatoes, and they love green bell peppers. Whatever it is, my students are doing taste tests and finding that so that they will play their monkey-based memory game optimally. So what is this memory game, and what do I want to look at? Well, the task that I'll tell you about is a task that I was particularly interested in. And that is a task where you have to form new associations online. We do this all the time. Name-face associations, meeting somebody for the first time. Professor Lip, I've never met you before, but now when I see you on the street, I will recognize you as that, and that's because something changed in my brain. So you've changed something in my brain. I will now recognize your face, and I associate that with your name. So we do that all the time. We know that's dependent on the hippocampus, because if I did not have my hippocampus, I would not be able to pull up that name when I meet him on the street tomorrow. And so I wanted to ask that same kind of question in monkeys. What if I ask monkeys to form new associations, and I ask, how do hippocampal neurons respond as animals are learning new associations for the first time? So let me show you how I got animals to learn new associations. So the question is, how do neurons in the hippocampus signal new associative memory? And so imagine you're a monkey in front of our monkey computer screens, and I found your superfood, which might be whatever, burritos or whatever you're having today. I don't know if that's your superfood, but at least it's a reward. I'll ask you to fixate this central fixation point. Then I'm going to show you a big complex image on the screen, and superimposed on that image are 4 targets: north, south, east, and west. All you have to do here is maintain fixation, but you have to pay attention to what's on the screen. So you have to remember that it's the gorilla picture for this trial. Then during a delay interval, I take that picture away, and, but the targets remain on the computer screen. And then what your job is to learn, with trial and error, this trial and other trials, which of these targets will give you a nice big drop of your favorite super juice when you move your eye to the target. When I extinguish that fixation point, that's your cue to move your eyes, and maybe you move south, and too bad, you don't get any super juice on that trial. But with trial and error, you learn that when that a gorilla is shown, you have to move your eyes to the north. And every time you move your eyes to the north for that trial, you get a big drop of your favorite juice. I'm going to ask you to learn 2 to 4 new picture target associations. They're going to be associated, each picture is going to be associated with a different target. And you do that gradually online. It takes you about 13 tries each if you're a monkey. You'll be very fast if you're a human, as you are. You do that intermix, and then I'm also going to give you what we call reference scenes. And these are pictures, these 4 pictures are very, very well known to the animal. And they know exactly which target is associated with reward. And you do that very well. So we're going to do both of these. I'm going to show you this is going to be an important control experiment. But the main question is, what's happening during learning of these new scenes? Okay, so they're learning to associate a picture with a motor response or a spatial location. A conditional, it's called conditional motor association learning, a picture response associative learning task. So first I'm going to ask, let's just show you a hippocampal cell and give you a feeling for how the cell responds during this trial. And the cell gives responses in units called action potentials or spikes. They're very brief changes in the membrane potential of the individual neurons. And we can count and quantify those action potentials or spikes. And so I'm going to tell you how many spikes per second I see during the time course of the trial, during the baseline period, which is 300 milliseconds, the scene period, 500 milliseconds, delay period, 700 milliseconds, and then this is when the eye movement is triggered. And what I'm showing you is for one hippocampal cell, I'm showing you the pattern that this cell fires for 3 different visual scenes. So the red is the average over all trials that day whenever the gorilla scene was shown. So you can see this cell likes, it starts to respond at the end of the scene, really high during the delay, and it comes back down. But you could also see that compared to the gorilla scene, responses to two other scenes, let's say this is a Washington Square, picture of Washington Square, and this one is a picture of the dental school. These two scenes do not elicit a lot of activity in the cell. And this cell really likes the response to respond whenever this gorilla scene is shown on the trial. So that's fun. We know that the cell is responsive to the task. But now the key question is, does it change? What happens during learning? Not averaged altogether, but what happens early when the animal doesn't really know the scene? And how does it change late? Or does it change at all? This particular cell. So that's what we're going to look at in the next slide. So here, first I'm showing you the learning curve. There is a learning curve. Animals learn this. At first, they perform very, very poorly on this trial, and then this learning curve is going up to 100%, and by the end of the session, this is 55 trials the animals completed, you see perfect performance here. This is just an estimation of a learning curve based on a dynamic logistic regression algorithm, but to show the real data, here are the errors in red dots and correct in blue dot performance on every single one of these 55 trials. So this is how it translates into the learning curve. You might wonder why the animal's performing so poorly here. There's 4 targets. How come he's performing less than 25% correct, which is chance? And the answer is that they have a not optimal strategy, these monkeys. So what typically happened when the animals starting to learn these new associations, they know at the beginning of the session, they're very familiar with these sessions, that, this is the hard part, I have to figure things out. And suddenly they get one right by chance, right? Maybe it's the south. And they say, aha, south. So for every new scene that comes up, they go south, 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 south. So that's what he was doing. He was getting it wrong. But then they figure it out. And slowly they come through. And suddenly he had this aha moment. I mean, look at this shift. That's good, right? You want your students learning like this. So what's happening? What's happening in the brain? So let me superimpose on this behavioral learning curve neural activity in these units, action potentials per second. This is speed of response of this neuron. And I'm going to first show you what happens in the first, let's say, 18 trials or so, when clearly, behaviorally, the animal has learned nothing. Okay, he does not know the association. And I'm going to show you the activity during the trial relative to baseline activity during the baseline period. And we can see that during this period, the cell is firing a little bit above baseline, but it's not changing at all during these first 18 trials. It's just kind of popping along like that, no change. But the key question is, what happens here? Here, something behaviorally happens. You have learned something here. That's why we use this test, so we can identify a point of learning. And here I've given you a very dramatic point of learning. So the question is, what happens with this white line, which is the neural activity in my example hippocampal cell? Well, what we see is that in parallel with this dramatic change in behavior, this dramatic change in learning, we have an equally dramatic change in neural activity that parallels the behavioral activity. Now, we started seeing this, and we didn't know what was going on at first. We didn't know exactly what we were going to see. But as we got sensitive that these cells were in that population that we're recording, you can be sitting there in the recording room, having the animal working and learning these things, and you could actually hear them by ear. You can start to hear these cells that get higher and higher on a given trial with learning. These are what we call changing cells. This cell is only signaling learning for this particular scene. The animals learned between 1 and 3 other scenes that day, no change in activity, or sometimes no response to those other associations being learned. Only a change in one association. And so we thought that was specific for learning. Very, very exciting. But there's another thing. Here, the animal is making a response to the north, because that's the correct response. And here, he's almost never making a response to the north. So maybe this is, I'm about to go north cell, or I want to go north cell, which isn't necessarily a learning cell, it's a motor cell. So that's why I told you about those control trials in which we had the animals perform the task, but they got it right every single time. So I can look at the response of the same cell to a different trial in which the animal is making the same response, but all the time. So if this is a I'm going to go north cell, then we should see very high activity to that reference scene where the animal is making the exact same movement to the north, but he learned that association. Instead, you see this activity. The same cell is responding not at all to that reference scene. So this is not a motor-related cell. This is a learning-related cell. Yes?
Where in the brain is the activity measured?
The activity is measured within or just adjacent to, these are extracellular measurements of cells in the hippocampus, neurons in the hippocampus. Any other questions? People follow me? I jumped quickly from the human neuropsychology to the electrophysiology, but hopefully, yes?
Is this a typical pattern that you would have with, let's say, 2 correct, and then it goes?
This is a dramatic one. They were pretty steep, these learning curves, but sometimes there was more correct here before you really went up. So they were a little bit, they were variable in their steepness, but they were on the steep side. So there was pretty fast learning in these particular associations, yes. Yes?
Do individual monkeys get better at similar tests?
Yeah, do individual monkeys get better at similar tasks? They absolutely get better, and I should say that we didn't start recording in these monkeys until they can learn very consistently multiple new associations a day. So they were good at it, and they absolutely got better. I mean, training a monkey to sit in front of a monitor and play a game like this for 2 or 3 hours, which is the length of this recording session, is kind of like training a 3-year-old to sit in front of a TV still for 3 hours. So it takes incremental training. That's why we need these superfoods and motivation for the animals. Any other questions? Okay, great. So we have these signals, learning signals in the brain, and you might wonder how prevalent are they? Recording in the hippocampus, we have a single electrode down there. Now we have a tetrode that we could record 5 to 10 cells at the same time. How many of the cells in the hippocampus are giving these kinds of signals? And the answer to that is about 20% of all the cells that we're able to isolate give these kind of changing signals like this. It's not all the cells. You wouldn't expect your entire hippocampus to suddenly want to do this task, but we get 20% of the cells that do that. And you have to remember that this is 20% of the cells that we can isolate. I already have bias because it's easier to isolate larger cells than smaller cells, but still within the cells that we attempt to isolate, about 20% are given these kinds of responses. I showed you one category of changing cells, these cells that go up with learning and stay up for as long as the animal performed the task. Those are called sustained changing cells. And I'm going to show you the second category of cells we identified. We called them baseline sustained changing cells. Here's sustained. And here's baseline sustained. These cells start out responding very well to a particular stimulus, let's say the gorilla scene. This is in a completely different recording session, a completely different cell. And these cells signal learning by going back down to baseline. This is significantly anti-correlated with the learning curve for this particular scene. This activity is not correlated with either positively or negatively with the learning curves of any other scene that the animal learned that day. So what do we think is happening? We think there's a tuning happening within the hippocampus, that as animals learn, some cells are coming up and becoming much more sensitive to that learned scene. Those are the sustained changing cells. And other cells are tuning down and becoming less sensitive to the stimulus itself. And those are the baseline sustained changing cells. So a dynamic process, we think, is going on. But we have kind of a picture to hang that on. Now, what exactly is happening in this hippocampus, this structure important for episodic memory? We see dynamic changes that happen in parallel with learning. But you might ask, well, Wendy, this neural activity is changing a little bit after after behavior has gone up. How can this neural activity drive behavior if it's happening later? And so the timing of these signals was always a critical question. And of course, our hypothesis was that any changes in the hippocampus are actually driving the behavior because in this task, when you lesion the hippocampus, you get a deficit in learning. You get a significantly slowed learning. But still, it's hard to interpret directly from the behavioral effects of lesion studies into neurophysiology. So that's why we do these studies. And so to address this question, we did a timing analysis. We did an estimation of the trial number of behavioral change as a function of the trial number of the neural change in these neurons that are changing with learning. And we asked, overall in the hippocampus, are the neurons changing after learning, behavioral learning, as we show here, or are some other neurons changing before learning? And what we found was, I'm going to plot this for every changing cell. I'm going to show you, I'm going to plot a single point showing you the behavioral change, the trial number of behavioral change as a function of the trial number of neural change. And all the dots down here are neurons that change before learning, and all the dots that will be up here are neurons that change after learning. And what we found was this. Pretty much an even distribution of neurons that change before and after learning. That suggests that fully half of the hippocampal neurons are changing at a time point that could help drive behavior. These other ones might be coming online, perhaps to help strengthen memories, because these memories are going up, and we certainly know our own associative memories. If you meet somebody new, that repetition is critical, that it's getting better and better and easier, and you may fail at first to remember the name of somebody that you really want to remember, but with repetition, that's coming through. And this might be the cells that help you strengthen those memories with repetition, these cells that are changing a little bit later. Okay, so that is a flavor of the kinds of studies and the ways in which we are looking at the real brain basis of memory formation. You might, yes?
Do you think that there could be different cells that are sort of the figuring it out cells versus once you figured it out, then you just remember what you figured out?
Yeah, so I think these play part of the figuring it out cells. And what I think is happening, this is not just me, but this is a lot of previous results. So once you know it, once you know an association, it's no longer dependent on the hippocampus. That's why HM could remember his mother and remember his school friends. And so what we know, what we believe, is that at first these new associations, new memories, will engage the hippocampus, and we'll see activity like this. But very well learned associations, I see activity like this. It's no longer dependent on the hippocampus. It's dependent on something else. I have it recorded in the area. So it's a transfer of information. First, it's dependent on the hippocampus, and then what we believe it moves out to cortical areas to be stored long term. This gets back to Alzheimer's disease. So early on in Alzheimer's disease, you get HM-like deficits. You have a poor ability to put new things in your long-term memory because Alzheimer's attacks the hippocampus in related areas. In fact, the very first area it attacks is the entorhinal cortex, which I mentioned. But then what it does is it spreads out into your cortex, and that's when you start getting more of the dementia, and you start getting the loss of your long-term memories. And that's because we think it does merge or migrate out of the hippocampus. Yes?
Would you expect audio-associated memory to have the same kind of pattern as vision-associated memory formation?
That's a good question. Do other kinds of modalities have the same kind of association? That's a good question, and it's hard to answer because vision has been studied most effectively in monkeys, Why? Because monkeys learn visual things most easily, just like we learn visual things most easily. But we certainly know from the anatomy that other sensory modalities get into the hippocampus, as they do in us. And we certainly know we can form more than just visual memories. But do I have the hard evidence that it's happening in the same way? No, because somatosensory memories are harder, and it's just harder. I chose this task because they can learn it. And if it takes them 5 days to learn, it's hard to study that thing. I suspect that it's going to be the same, and if I was able to come up with an easy, multimodal, non-visual task to do it, that it would be the same. But I don't have the data to back that up. That's my guess. Did you have a question? No. Back there, yes.
Did HM have the ability to input and retrieve music, lyrics, and melody?
No. He could not. They tested basically all modalities possible. All new information that is encoded in an episodic kind of way was not available to him. There was one famous study where they looked at, they tested him on things that came into the culture after he became amnesic. And there were just a few things that he actually remembered. He knew Princess Diana. He could recognize that name, and he watched a lot of TV. So there were a few things that came into his memory, but we believe that they came in by different means. It was probably a very rote kind of slow learning memory that our brain is still capable of, that he was able to exhibit. Yes?
It's obvious that the long-term memory is not in the hippocampus, but
yeah.
Is there a way of training other parts of the brain somewhat to do the job of the hippocampus? And then,
unfortunately, the answer is no. The hippocampus is quite special, and so, well, yes and no. It depends how strict you are in your determination of like the hippocampus. You can learn things. You learn things very slowly, with not a lot of flexibility, and very rigidly. And that's because another brain system takes over, and that brain system is often the striatal system, which is a motor learning, kind of skill learning pathway. So you can get around things, certain things, much more slowly. But if you want that fast, flexible kind of memory where I can not only remember this room, but I can remember the configuration, and that you're wearing black and you're wearing black and you introduced me and you're sitting over there. That's the kind of flexibility and richness that the hippocampus brings to all of our experiences that we're kind of taking in our everyday lives. Yes.
What was the age category of the animals that were tested?
These were adults, young adults, so they're equivalent to adults and young adults. Any other questions?
Yeah.
Yes.
So for patients with amnestic disorders,
yeah.
Do you have any familiarity with pharmacological agents that can improve memory, even if it's over an abbreviated period?
Yeah. Do I have any familiarity with pharmacological agents that can improve memory in people that are amnesic? It all depends on what kind of amnesia you have, and all of the data that I've read is that the best you get is a mild improvement, and I'm talking mainly about the drugs available for Alzheimer's disease and the dementia associated with that. It's a little bit. I mean, these are drugs that help particular neurotransmitter systems that I'm not going to talk about today, that can give a little boost, but they're not replacing these cells. I mean, how are you going to replace these kinds of cells if they die in Alzheimer's disease? That is a hard problem. You have to go in and stop that disease from happening and preserve the rest of the healthy cells that can continue to do this, because once these are gone, it's very hard from the outside to do anything, unfortunately. Any other questions? Yes?
They've shown that neurons in the other parts of the brain multiply. It's not that when you lose them. You get more of them if you study and learn. So is there any way of let's say you're losing half of the hippocampus? Is there any way of giving the other side a little bit more potential? Well,
so let's be clear about this. There are two parts of the brain that we know generate new neurons through adulthood. It's not the whole brain. It's only two parts of the brain. One of those two parts is the hippocampus. So once you lose that in Alzheimer's, that part that was growing new cells, it's gone. The other part is the olfactory bulb, which we don't understand why, at least humans are not very good at olfactory, and we don't use olfactory cues as much as, say, a rodent. And so yes, you still retain that, but more olfactory neurons are not going to help you in your everyday cognitive life that much. Now, what you might be referring to is studies, very exciting studies, that it's a good precursor for the next part of the talk, that showed that if you exercise for a year, these are 65 people that are 65 or older, you can actually increase the size. It's actually increased the size of your hippocampus significantly. But other studies have shown that other brain areas are, white matter bundles are growing. That's not new neurons. It's other things that are growing. It's mainly support cells in the brain that could be helpful, but it's not the new neurons that multiply. So it's, we're kind of blocked at all these possibilities, especially with age-related cognitive decline in the hippocampus and Alzheimer's disease. Your hippocampus is special. You want to preserve it. And so the question is, how do I improve my memory? How do I improve my hippocampal function? And so this was a question that I got very interested in because I noticed that one day, or not one day, but over a couple of weeks, that my writing, I'm sure you guys have to write grants. I spent a lot of time writing grants. And I noticed that my grant writing was getting easier. I could make associations better. Things were moving more smoothly. Ideas were coming together better. It's like, what is going on here? And it would corresponded to a time where I started going to the gym more regularly, and I got very, very addicted to this one class at the gym. The class at the gym was called Intensati. It was developed by a fitness instructor here in New York City named Patricia Moreno, and I just love this workout. It is unique because it's an aerobic workout. We take physical movements from kickboxing and dance and yoga, which is like a lot of other aerobic kinds of classes. But the unique thing is that we pair every movement with a positive spoken affirmation. So there's an interesting cognitive component to it. So you're punching like you would in kickboxing class, but in this class, you actually have to yell out, "I am strong now." So once you get over the weirdness of actually yelling out in class, it's actually a lot of fun, and it actually does something very, very striking. It actually shifts your thoughts from typical thoughts of, "I'm hungry. What's for dinner, after this workout? How much longer is it?" to, "I am strong. I believe I will succeed." All the positive things that we do, and the affirmations aren't the same every week. There are themes that we do, and we change them every month. And so there was an interesting memory component. You have to remember what these affirmations are. You're saying them out loud. You're increasing your cardio output when you say these affirmations, and you're learning all these kind of dance cardio movements all at the same time. And I thought it was so much fun. I started going very, very regularly. And I noticed that when I did this, it really helped my writing. I thought, I want to learn more about this. And so the first idea is teaching and learning seminar is the best way to learn something is to teach an undergraduate class about it. So I decided I wanted to learn the literature. It was far enough away. And so I went back and I started developing this new undergraduate course called Can Exercise Change Your Brain? And I was going to review all of the animal literature and the human literature on the effects of exercise on the brain. So that was going to be a fun course, and hopefully I was going to attract my neuroscience majors at downtown campus. And it turns out that the precursors for the studies were very, very familiar. They came from studies done in the 1960s by Miriam Diamond, Mark Rosenzweig, and their colleagues at UC Berkeley. And what Diamond Rosenzweig did way back in the 60s is they were interested in the effects of the environment on the brain. How can the environment change the brain? So they did this by raising rats in what they called enriched environments. So enriched environments meant more rats living together in a big space with lots of toys. So they have ladders, they have swings, and they played around. And they compared the brains of these rats to other rats that they lived in what they call the impoverished environment, just one other rat in there with no toys. What they found was striking changes in the brain. They found, what they did is they measured cortical thickness here. And cortical thickness was increased in the rats that lived in the enriched environment, in the visual cortex, in the somatosensory cortex, and in the motor cortex. Why? Because they imagined that there was a lot more visual stimulation with other rats to look at, other toys to play with. Somatosensory, they were jostling around. They were getting a lot more somatosensory activation than they were in this empty cage with maybe one other rat and no toys. And motor cortex, because they were running around a lot more. So their cortex was actually thicker. The brain grew. And let me just clarify, it's not the neurons that grew, it was the glia cells that grew, but the thickness was bigger. They found neurotransmitter levels changed in the brains that were raised in enriched environments compared to the impoverished environments. They found more synapses. So while there's not more neurons, there were more connections between the neurons that were there. Angiogenesis, the birth of new or the growth of new blood vessels. Why are blood vessels important? Well, the brain is, as you know, the #1 user of oxygen in the body. The more ability the brain has to get that oxygen, the better it can work. So you actually generate, with this enriched environment, more blood vessels. And here's actually a nice blood vessel. You can see them throughout the cortex in the rat. Growth factors increase dramatically when you get erased in an enriched environment. And the cool thing that was not found in the 60s but much later is that enriched environments activate what we call hippocampal neurogenesis, the birth of new neurons in the hippocampus. We know this happens normally throughout adulthood. Hippocampal neurons are normally born, and they're born in this subregion of the hippocampus right here called the dentate gyrus. What raising in enriched environments does is it enhances that hippocampal neurogenesis, so you get even more neurons. So about now you should be thinking, how can I get enriched? What can enrich me? You know, it's pretty easy to enrich a rat. Can I get enriched? Because I want all this happening in my brain. Well, the question is, one of the questions they asked next is, there's lots of complex things happening in this enriched environment. What is causing all those great brain changes? Is it the social interaction? We're very social animals, so maybe it's just that I got to interact more with my friends. Was it the game playing, which is related to social thing? Was it the running around? They had exercise wheels, and they naturally kind of ran around, chased each other more. What was actually doing it? And they did lots of clever controls. They even, I always thought this was my favorite control, is they did what they called the TV watching control. They compared rats living in the enriched environment to other rats that just could watch the enriched environment. They couldn't play. It was kind of sad. So it's like watching TV. Those TV watching rats or enriched environment watching rats, nothing in their brain changed. But these rats still had brain changes. So what element was it about this complex environment? And the answer? Exercise. You can get all these changes just with exercise. Just all you have to do in a rat, is give them access to this. Doesn't matter if the other toys are there. It doesn't matter whether all the other rats are there. All he needs is to run here, and he gets all those changes. Okay, so that was cool, and that was going to be like the major message of this, all of this research that I was doing to look at the history of how we know all this stuff in going back to my undergraduate class. But the other thing is that the whole idea behind the class was inspired by exercise in the first place. So I thought, wouldn't it be fun to actually bring exercise into the classroom and kind of let the students experience exercise as we're learning about the effects of exercise on the brain? So I did something a little bit unique. And so I actually got trained to be an Intensati instructor because I loved that workout. And what I decided to do is I realized that these students were not just forcing me to learn the literature, but they can be the subjects in my first experimental study. All I need to do was to test them cognitively at the beginning, at the end of the semester, and what I was going to do anyway was work out with them for an hour before the class where I told them about what exercise was doing to their brain. So that's exactly what we did. And that turned into my Can Exercise Change Your Brain class for upper division undergraduates, neuroscience majors. I've taught it twice so far, fall of 2009 and spring of 2011. And I'm going to tell you what the results are. Could I see hippocampal improvement with a semester-long increase in exercise in my healthy young neuroscience majors? My collaborators on this project were two neurologists up at Columbia, Adam Brinkman and Scott Small, and they brought something very, very important to the study. They brought the task that I needed, that they suspected would be sensitive to this study. What task was this? Well, the hypothesis was that my students and myself were like rats, and because we were exercising more, we would have increased hippocampal neurogenesis, and so I would have more cells in my dentate gyrus. And so I needed a task that was sensitive to dentate gyrus function. And Scott and Adam had that task. It was a Benton visual recognition task, and I'm going to show you it in a second. And they showed in imaging studies that the dentate gyrus is highly active when normal people are performing this task, but not when it's performing. Dentate gyrus does not activate for a different kind of memory task, delayed retention. And then the entorhinal cortex has the opposite pattern. But the important data is right here. Dentate gyrus has large and specific activity to this particular task. So maybe I would see even better activity and better performance on this task in my students that exercised relative to other students that did not exercise during the semester. So what is this task? This task is basically a memory encoding task. It's quite challenging. We show them a complex geometrical figure, and after a very short delay, I show you these two, and I ask, which one did you see? Is it the one on the right or the one on the left? Which one is it? Very good. So you have excellent dentate gyri. And so there were a whole bunch of these kinds of tasks that we had the students perform before and after learning. So cognitive testing before, including that Benton recognition task. We had a whole semester of 1 hour of exercise plus lecture and discussion about the effects of all the literature. We had cognitive testing at the end, and then final papers. And then I cajoled one of my colleagues who was teaching a parallel neuroscience elective class with the same timing to let me test his class cognitively at the beginning, at the end of the semester, and these were my control subjects. So what did we find? Did my students improve their memory with exercise? And the answer was interesting. So first, here is the % correct performance on that Benton recognition task. Performance scores are here. Here's the performance of controls before the beginning of the semester, the end of the semester. Here's the performance of exercise. Exercise performance didn't change. But what was striking here is look at these controls. How come they're doing so much worse at the end of the semester? I don't know about you. Do your dental students get stressed during finals? Yeah. Oh, yeah. Here are our NYU undergraduates getting very stressed at finals. I couldn't help it. This was the design of my task. I had to test them at the end of the semester. This was the time when they were preparing for finals. And what we found is a detriment in their performance of the control group. And so what we might be seeing here is not necessarily an improvement, but perhaps a protection from stress. I don't know about you, but I would love to be protected from stress. So this was one of our first exciting findings. This is still preliminary. We need to do a larger study and hopefully not get away, or not get away from, but study the effects of stress in a more controlled way, not just, oh, I have to teach you, I have to test you at finals, so we get stress along with that. my students got significantly better. This is a significant improvement in the reaction times. This is reaction times are decreasing, which is better. This is the exercise group. So my class did get significantly faster at responding correctly to this class, and they seem to be protected from stress. So the first studies, interesting because most of the previous study has focused on elderly people where their cognition is declining. So the next question, 2 more studies just to go over on the exercise front. What about just going for an hour? I don't know if you have time to jump into a 15-week semester long increase in exercise, but you probably have time to go to the gym for an hour if you really, really try. So what is the effect of 1 hour of aerobic exercise? This is again in neuroscience, undergraduates, NYU undergraduates. I'm not going to tell you about the EEG studies. We're using the same Benton visual recognition task from them as well as other tasks. And then these were our EEG colleagues, but I'm not going to focus on that. What we had here was a larger study. We had 50 people in the exercise group, 50 people in the control group. We got cognitive testing. We did exercise with them. This was not intensati. This was exercise bike, and then either 30, 60, 90, or 120 minutes after their workout, they came back and they were cognitively tested again. Our control group was they couldn't actively go on the bike, but they watched Jack Bauer jump around for an hour on the TV show 24. And so that was our control, TV watching. They TV watched in the gym where these guys were riding their bikes, so they ride their bike and they watched TV. Everything else was identical. So what did we find? Well, first let me show you what we tested. General tests of reading abilities, just to make sure that their reading was fine. We focused on tests of attention. Stroop color word test is a task where you have a list of words. All the words are words that name colors, and the words are either printed in black or in the same color as the word or a different color of the word. And it's actually very hard. On command, you either have to read the word, read the color of the word, or read the color of the word. And it's shifting attention task. Trail making is also shifting attention. Symbol-digit modalities is symbol-digit modalities is shifting attention. Trail making is kind of spatial paying attention. Oral word association is a verbal memory task. Digit span, short-term memory task. And Hopkins verbal learning task is a task where it's basically a recall task. It's very sensitive to Alzheimer's disease. You have a list of 12 words. I give it to you. Then you just have to immediately tell me all the words you remember, and then tell me again 20 minutes later. Very sensitive to early Alzheimer's disease. So what do we see? How does exercise for just an hour affect performance on this task? Well, we got 3 different results. The first was on that Hopkins verbal learning test, that recall test sensitive to Alzheimer's disease. But the results were interesting. What we got was maybe a little improvement. This is immediate recall and delayed. Little improvement on exercise. But look what's happening to controls. Controls are getting worse after their TV watching. So I think inadvertently we found terrible effects of TV watching on regular recall. And what we think is happening is there's a lot of interference happening. You're remembering what Jack Bauer was doing and remember all the storylines, and it makes it harder for you to focus on and recall those 12 words. So, this is might be more of an effect of the TV watching than the exercise. But we have 2 more interesting results. One is on that Stroop test, and one is the, again, attention-shifting symbol-digit modalities. Here, the controls get a little bit better the second time, as you would suspect. So TV watching is not making their performance work. And exercise makes it even better. So here, what we're seeing is clear evidence of improved attention functions after an hour of exercise. And I'm sure you appreciate that. People come out of exercise, they feel better. They feel like I can focus on things better. And here we're just quantifying that and showing that statistically there are improved memory functions after just an hour of exercise. So we have both long-term and acute aerobic exercise having significant positive effects on cognitive function. Different durations of exercise may have different effects. We had the kind of Benton visual recognition improvements with my long-term exercise, and frontal improvements after just an hour of aerobic exercise when they're tested immediately after. And related to what you're interested in, these findings have implications for education. So one of my long-term goals is to find the prescription for exercise to help my students actually encode better, to learn better, to perform better on tasks. What do they have to do? Do they have to exercise 6 times a week and really become, you know, a marathon runner? No, I don't think that's the case. But what is the most strategic, least time-consuming way that they can optimize the way that they study, the way that they either memorize or understand conceptual things? Those are some of the questions that I want to get towards. Yes?
Do you think that, I mean, I know it's preliminary, but do you think that this effect might, over time, it might become less stark? I mean, you know, as they're exercising more and more and going to your classes, do you think that you might see?
Yeah, is there a max? Right. Is there a max? And there could be. But really only elite athletes have reached that. And that's, I mean, I'm fascinated. I would love to get a group of ultra marathon runners in and see what the effect is. But we're typically working with a bunch of sedentary people that could use exercise for more than just cognitive function. And so there's lots of reasons to continue this. Okay, so lastly, I'll just say that I'm particularly excited about the last direction, which is the effects of exercise in neurological conditions like traumatic brain injury. And when we did a similar 8-week study of exercise in traumatic brain injury subjects, the exciting results are that our TBI subjects reported significantly less depressive symptoms, more positive affect. They got faster on that visual recognition task, and the ones that completed the most exercise sessions reported better self-esteem. So there are not only cognitive effects, but mood effects that are very, very relevant, particularly to TBI subjects. So lots of interesting areas, and I would love to combine studies of the hippocampus and physiology of hippocampus with my studies of the effects of exercise in memory in the future. And thank you for your attention.